Hi everyone, welcome to my full course in genome-wide association study, genome selection and quantitative genetics. My name is Yanjun Zhang and I'm a postdoc from Swedish University of Agricultural Science. I started my PhD in 2014 on this topic, and after I finished my PhD, I did 3 years postdoc on this topic. When I was learning this topic, I find it very difficult to find a comprehensive lectures on this topic, and I sort of pieced together bits of information from websites, YouTubes, and my colleagues, and books and courses I took. I've been wanting to put up such a session for a very long time. With the pandemic, I'm stuck in working at home. The bars are closed, the gym is closed, and it seems it's not going to be open in the near future, so I'm doing this right now. So before we start, I'll give you a brief history of quantitative genetics. It's really going to be a brief history. Darwin formed evolutionary genetics by studying the similarities and the differences among species, and later on, Mendel found the Mendelian inheritance law, where quantitative genetics started to divert from the evolutionary genetics framework, focusing on studying the within species similarities. Later on, Galton coined the term heritability when he was studying the similarity of human head between parents and offspring and invented linear regression. Fisher summarized all those achievements and come up with a unified framework, and that's when quantitative genetics officially became an independent subject. Since then, a lot of exciting findings has been discovered. QTL mappings. From a limited number of molecular markers, we're moving into the next generation sequencing, and we can look on the whole genome variations and associated to phenotypic variations. A lot more bigger project has been invented, both in humans, in plants, and in animals. Before we start to talk about GWAS and GS, there's a lot of basics to cover. I'll start by introducing what is a chromosome and what is a gene, and what is a phenotype and how variation in genes can affect the phenotypes. Then I'll talk about what is molecular markers and what marker has been used in old days, and the progression of technologies in quantify the molecular marker, including the SNP array, the next generation sequencing, and the whole world derived techniques. It's not possible to really cover all of those without talking about statistics, but this is not a course specifically focused on stats, and I try to minimize the stats into the minimal that to get you going. I'll be introducing what is a variable, what is linear regression about, and what are the effect sizes and p-values means, and what is really multiple testing, how is that going to affect our GWAS results and analysis, and how do we use linear algebra to describe the problems that we see in GWAS and GS. I'll also give a little bit about the theory on how to estimate the effect sizes and p-value and other parameters that we encountered along the process, including likelihood and Bayesian statistics. Then we're moving into more quantitative genetics basis. I'll talk about study designs. You probably heard about F2 and heard about backcrossing or advanced intercross line or just wild natural populations or the breeding materials. What are the similarity and differences in terms of population structure, the LD structure, how haplotype structure is going to affect the analysis we're going to do. And next, I'll talk a little bit about bioinformatics basis. Today, the amount of data we encounter in typical GWAS is way beyond what we can handle just by handwriting. So we're going to use a lot of tools and a little bit of bioinformatics skills. I'll cover what is reference genome and how do we call the variant we use in the GWAS analysis, and what kind of type of file format we are going to encounter, and how to transfer between file formats, and what kind of software and tools are we going to use. Then I'll talk a little bit about handling genotype imputation, because that's something you might encounter in the future projects. Then we move on to the quantitative traits. It's really the simple concept, but I find it very easy to get started, but it's very difficult to have a conceptual and deeper understanding on that. The most familiar traits probably is human head. It's a continuous distribution, meaning that we have people ranging from 1.x meter to all the way up 2 meter and follows bell-shaped distribution. And we also have categorical data set like the stage 1 cancer, stage 2 for cancer, or even something arbitrarily defined like hair. It's a dark color or it's a white color. All those measurements of the quantitative traits are arbitrary and humanly defined. So it has to be put into biological context. Then I'll talk about heritabilities, why that is really important parameter to think about when we do genome-wide association studies, genomic selection. And then I'll talk about the genetic architecture. What do we see and what do we mean by genetic architecture or genetic basis of quantitative traits? Then we will talk about QTL mappings and linkage maps. We'll talk about typical QTL mapping study designs and how do we handle the genotype and phenotype quality control in terms of missing rate, Heidelberg, and segregation distortions, and how we're using map plans to see whether we did a good job in the genotyping and in the linkage map construction. It's really a long way to get to GWAS, which is the focus of this course. I'll first start to introducing how we do GWAS, the process and diagnosis, what is QQ plot about, and what is inflation, and what is population structure. Then I talk about several ways of accounting for population structure, either using genomic control or using PCA or linear mixed model, or a combination of those techniques. Then I'll cover what does kinship means and how does it really relate to population structure, why that would really works and serving as a workhorse in the GWAS and GS. Once we have a few candidate associations or SNPs, I'll talk about how to estimate variance explained by a single variant or a set of variants. How is that estimated to ask how much heritability we are missing? After I walk you through the process of how to do GWAS and how to do diagnostics, I'll talk about the assumptions we have been made. Some of those assumptions are very arbitrary, and it's going to have a bigger impact on the false negatives and the false positives we talked about. Then I'll give you a little bit of extension on the GWAS. In many of the projects we do, the phenotype is continuous, but very often you might find you're in a situation where the phenotype is binary, and how we really account for that, how heritability estimates change, interpretation of the effect sizes also change. And there are also other types of study design where you chop off the population and only genotype the top and bottom quantile of the population. That's the top tail design, how we really interpret the result from there. And other times you will find non-parametric GWAS, what we talk about. Very rare, but could be very useful when we have a categorical trait and we don't want to put heavy assumptions on that. From here, we know how to do a GWAS, what the background is, and once we have done the GWAS, very often that we find a very broad peak. I mean, a peak might cover several kV or several megabit, where there's lots of genes there. In this situation, we have to use in a statistical task called conditional task to find a representative variant from a broad peak. And how do we aggregate the summary statistic from the broad peak to pinpoint the genes, and how do we refine the analysis using haplotype, because the smallest unit inherited between generations is really haplotype rather than the variant we are using to do GWAS. Then I talk about mathematically how GWAS and GS is really related, and what is a Breeder equation, how we use that to predicting the selection response from one generation to another. There are two types of methods basically in the GWAS and the GS framework. One is likelihoodist that I am more familiar with, but there's other type of method called Bayesian framework. Technically, Bayesian framework relaxes a lot of the heavy assumptions that were made in the likelihood framework. It gives slightly better performance when it comes to prediction. So far, we have been focusing on QTLs that regulating the mean of the phenotype. There's another aspect to it, is the variance of phenotype. That is to say, the QTL regulating the variance of the phenotype rather than the mean. This is something a professor in my PhD group have started to do and turned to be broadly applicable to other fields and species. I'll talk about the motivation underlying this and how do we really handle inflation in this. It's more difficult to handle inflation than the mean GWAS. And I'll give you a few examples on causes and cases, because very often the variants of a phenotype have a biological context, and there has been several studies that pinpoint the really interesting biology underlying this type of QTL. So far, we have been assuming QTLs combine their effects additively. That is to say, if you have two QTLs, one with 5 grams, the other with 10 grams, the combined effect will be 15 grams. But in reality, it's far more complex. We have what we call epistasis. That is a combination of allelic effect from two QTLs is beyond the single aggregation. And this is something our group has been focusing on for a very, very long time. Then there's dominance. At some time point of your study or research, you must have heard about heterosis. That is, the F1 hybrids outperform their parents. It's a very common phenomenon that broadly used in the animal breeding or crop breeding fields. Then I'll talk about genotype by-environment interaction. That describes the phenotype is really a manifest of the environment. Once the environment changes, the phenotype can change drastically. That's the field where I'm right now focusing a lot of energy on, and I'll talk about how plasticity, that is, individual being sensitive or less sensitive to the environment, and how do we go about on that. And so far we have been talking about using single traits. And actually, in the real world, one individual always have many traits, and once we do selection, we always do selection on multiple traits. For example, in crop breeding, we want a crop to have high yield. We also want it to be very efficient in nitrogen use. We also want it to be very good in disease resistant. So we are constantly handling multiple traits. I'll talk about how we use phenotypic or genetic correlation to describe the relation between two traits. And since the similarity between traits can come in from pleiotropy and linkage, I'll give an introduction on that. And how do we really distinguish pleiotropy from linkage once we have the genotype data? Once we do selection on one trait, we get a response and we can predict that using Breeder's equations. But once we do selection on one trait, we also get a correlated response from other traits. Because multiple traits are always being selected, it obeyes the predictions from single trait Breeder equation. That's why the multi-trait Breeder equation was developed, and we'll cover a little bit about that. We can use the multiple traits theory and apply that to the single trait across the environment and to study plasticity. As you might have heard, there are a lot of transcriptome data and methylomes and multiple sources of omics data. And there's a lot of development trying to using this molecular phenotype to infer a causation between those phenotypes. Methods including transcript-wide association studies, omics-wide association studies, Mendelian randomization. I'll talk about those topics and give a few examples and other types of machine learning methods. This is not a field I'm really good at, but I would like to give you a broader perspective in this field. If I missed something, please leave the comments below and we can improve it together. Then I'll talk about how we, just by looking at individual studies from multiple agencies trying to power up that information using meta-analysis. I'll talk a little bit about how to deal with the rare variance using burden test. And so far, we have only been focusing on doing GWAS using the linear model, linear mixed model world. There has been actually a lot more developments even within the linear mixed model framework. I'll start to give you a broader perspective on why these methods are come up, what assumptions they have actually relaxed, and why are they useful in a certain population or a certain setup. Then talk a little bit about a multi-locus model that I've been working a little bit with and a lot of people are using today, and the different similarities between likelihoodist and Bayesian frameworks and how they really perform in real data analysis. And then I'm generally talking about what really affects the detection power of the GWAS, and once we apply multiple methods in the same dataset, how do we deal with the agreement, or very often it's disagreement. Then we move on to the next chapters that's going to be more application or future development of this field, including how we have used GWAS to infer the genetic architecture and biological mechanisms underlying it, and how we can use in GWAS to infer the evolution history of a trait, or how a trait have evolved during domestication and artificial selection, or the range expansion in nature. And I'll talk about some of the ongoing developments in this field that I'm trying to use in the functional validation, functional annotation, fully integrating the genomics with genetics and how we better understand the GWAS hit that very often falls outside of the gene and come up with the regulatory mechanisms that might be very useful underlying the biology of the traits. Along those lines, the tissue specificity, because for multicellular species, we have multiple cells in the organs and tissues, and the expression pattern of the same genes varies across this, and how we have been using the tissue specificity to inform where this gene has functioned in what organ and tissue that lead to the variation of the phenotype. And then I'll talk about more in either human health or the crop breeding fields. How do we using GWAS to make an impact on the real world? After this is done, we will talk a little bit about the ongoing developments. I'll cover why genome assembly in some species is still challenging problems, and how we're moving from SNPs to the series or copy number variations or even haplotypes. And one ongoing aspect about this on this field is predicting the breeding design. That is to say, how do we really shuffle the genomes of the populations we have right now to enrich the favorable alleles in future generations that are going to give us a better variety than we can do right now. Then we will talk about, in general, how do we do prediction and diagnostics in using the omics data to improve our healthcare. And with this, I would like to conclude my introduction. If you like my video, please hit the like button and subscribe. Otherwise, I wish you all the best in the future study and research. Thank you for watching.